Thank you, Dr. Johnson. Truly excellent work advancing pharmacogenomics in a number of different therapeutic areas, right, in terms of depression as well as pain, and we all eagerly await those results. So I'm your next speaker, and it's really a pleasure to kick off and talk about everything we're doing here in Pittsburgh, specifically in pharmacogenomics. So a little bit about Pittsburgh and UPMC, University of Pittsburgh. We are a large integrated financial delivery system, which means we have a truly awesome opportunity to show the value of pharmacogenomic testing for large populations. And really leverages everything from the awesome research enterprise we have at the University of Pittsburgh all the way to our health plan, where we have payer data in large data sets on over 4 million covered lives to leverage this benefit, as well as excellent testing on site. The Pittsburgh Pharmacogenomics program is really focused in 3 areas. One is clinical, and that's our Precise RX program, which is making interventions at the point of care and at the bedside to be able to change care with clinical billing and capturing the results. Our Pharmacogenomics Center of Excellence, which I'll talk about in a moment, is a large population research study that's enrolling over 150,000 patients to show the value and to bring those discoveries back into the lab or from the lab back into practice. And then finally, education, where we understand that actually education is one of the primary barriers. So where did we start? We were really excited to learn from experts in the field at the very beginning and all the pilot projects that were being done to accelerate our own implementation here in Pittsburgh, focused in cardiovascular and making a simple intervention, or what seemed to be a simple intervention, and that was guiding antiplatelet therapy in the cardiac catheterization lab. So here's our press release in late 2015 when we launched, and through the last 5 years, we've continued doing standard of care genotyping. So if anyone has the unfortunate result of needing a stent here for an MI or PCI events here at the University of Pittsburgh at Presbyterian Hospital, we would encourage the use to direct therapy for antiplatelets using CYP2C19 testing. So as we move forward, you can see our cath rates, even through COVID, are remarkably consistent, and about 750 patients a year, approaching 4,000 patients of clinical genotyping. We can see about 1/3 of our patients are carrying loss of function variants, and about 1 in 5 overall we're making interventions. Clopidogrel or Plavix is still our workhorse. 3/4 of our patients are receiving therapy, and not only we're changing therapy when we perceive clopidogrel may not work based on the genetic guided strategy, but we're also starting to think about when we could step down and have perhaps a greater confidence that clopidogrel may work in patients that do not carry those variants, so a de-escalation therapy. On the right-hand side is data from the IGNITE network, certainly involving University of Florida you just heard about, and others, where we were able to pool data together and show the value of this intervention. So I won't go deep into this, but you can see the difference in the Kaplan-Meier curve for hard outcomes. So that's death, MI, or stroke on the y-axis. And very simply, if you compare patients who are carrying the variant that had a modified therapy in the blue line to those that were not modified for a variety of reasons, this was a clinician-directed strategy. There was a 2.2-fold difference in the hazard ratio between those 2 populations, showing the value, and there was no difference between the other curves, showing that with a precision medicine approach, we can lower the risk in a patient population carrying a variant to that was not carrying the variant by guided prescribing. So there's been tons of publications by this group, a highly productive pharmacogenomics working group that has shown the value, everything from economics to the add-on value of testing for additional drugs using the same single gene test. And now we've all started thinking about panels and preemptive testing, where we could use value on all the pharmacogenomic targets that Dr. Johnson just mentioned, as well as the ones that have high value beyond what's done in the IGNITE network. So how does that look in Pittsburgh? Well, we've created this Pharmacogenomic Center of Excellence in partnership with Thermo Fisher Scientific and all the great components of the Pitt and UPMC health system. So that is our Pitt Pharmacy, CTSI, the Institute for Precision Medicine, UPMC, our payer system, as well as our venture group, UPMC Enterprises. And we have committed and funded to achieve genotyping 150,000 patients using a panel-based test. And this project was launched in a phased development in 2018. You can see our hypotheses here. They are spanning away from research to clinical to the first one there, which is to truly drive research engagement. We believe in our collecting data to show this, that patients want this. They see it as a way of wanting to come into research. Our providers see the value of pharmacogenomics and are more excited to recruit into biobanks and bring people into research. And then obviously to improve medication outcomes where there's high value targets for pharmacogenomics and those gene drug pairs where there's clear CPIC recommendations as well as FDA recommendations. And then finally to really allow for enhanced genotype to phenotype discovery. Although there's some great data out there at the highest levels for pharmacogenomics, there's also thousands of associations that are just below that level that are just waiting for validation sets. So we need these large studies that can do that as long as we can collect both the phenotype, the drug exposure, and the genetics together. So that's what we intend to do. The model's pretty simple. Patients come in to the biobank, have elective return of results. I'll talk about how that's done in a moment. And then the samples and the data are made available to investigators here at Pitt and UPMC for able to make those new associations. The model for scaling is also seemingly simple, but I think elegant in that it allows us to get to high numbers with making all our individual stakeholders excited to participate. So recruitment drives value on the clinical side and on the research side, which drives further engagement and then further recruitment. What makes you want to go home to your families and say, that was a cool study, I'm interested, you should consider participating if you're interested? What makes our clinician partners who are already overwhelmed in the workforce in their own clinical care model saying, this has value for our patients? And the answer for both of those things at our institution seems to be pharmacogenomics. And so for that reason, we're really excited and we think there's an opportunity to scale quite large here and elsewhere. More discreetly, the model looks like this. Patients come in, they join our new CTSI P+Me Discovery Biobank. They provide a sample, which is delivered within chain of custody in a CAP-CLIA workflow to our UPMC Genome Center. That data flows off our array into our research database, where we're able to link with EHR data and health plan data. And then on the clinical side, as we understand evidence and clinically validate the panel for return, those results will flow out through our clinical pipeline to UPMC EHRs, both Epic and Cerner, across our entire health system, and to be made available to patients and their clinicians. So phases, this is a very large project, and these are the phases. Recruitment infrastructure and IRB accomplishments were the first thing out of the gate. Then to develop CAP-CLIA workflows, bring in equipment on site, and scale up those processes and data pipelines. Then think about everything we need to do in the EHRs, decision support, supporting our tens of thousands of providers here at UPMC and beyond. And then finally getting the return of results right to all those groups, especially to patients. Like in the All of Us program we heard earlier, they are our tools of engagement. They're our ultimate customers. And those are the ones that we want to make sure are excited about research participation. This is where we are in phase one, and I'm pretty sure everybody can guess what that flat line is. That is where we're not allowed to electively bring patients in for research-based recruitment. The cool part of that is we are ending that now, and we have come out of that now, and that what was 200 or 150, 200 patients a week for enrollment now is a lot greater in terms of numbers. In terms of remnant samples, we've added that into our pipeline, so we're able to get clinical remnants from patients that consent. We've moved to electronic consenting, embedded into workflows. So just like in the ADOPT study, we've started to think about where patients might use this data, and we specifically integrated into UPMC pre-op clinics. These are a great opportunity where patients come in very early before their procedures. We're able to start preemptive testing there within a simple workflow, have the results in hand before their surgeries, and then guide everything from pain to anticoagulation to anti-nausea and even depression therapies afterwards if it's necessary. Second part of phase one was thinking about the IRB model. We spent a lot of time getting this right, we think, specifically making sure we're able to share samples both internally and externally, that there are commercialization opportunities. There's a certificate of confidentiality that we can protect participants with. We have very broad data sharing, the ability to do ongoing whole genome analysis on top of this and deposit our data in national repositories, EHR linkages, recontact, and then return a result that's elective. So the elective, and highlighted there, is for elective return of pharmacogenomic targets. And we asked patients, or participants in this case, whether they want their results back personally and for us to put the results in their UPMC medical record. That's an and, not an or, but even without that consent part of it, we would be able to use the data for research purposes. So initially, we had a very, very high percentage. So I think it was 97% for the first few months when we got started. That number has settled down around 91 or so percent, which is as expected as we get more folks into the program. But we're really excited that that number is so high. These are patients coming in that say, I want my data back, and it reflects what we're seeing, what we're seeing in the community with increased interest in genomics in general. Phase 2 was establishing testing. So this is a picture of our UPMC Genome Center. And we have deployed a contemporary array, the Pharmacogenomics array that's developed by Thermo Fisher. And we're getting excellent yields for our DNA. So for investigators long term, we have on average 100 micrograms of DNA per sample, which we believe should be enough for lots of analyses long term. And we have developed a clinical test or clinical deliverable called the UPMC Comprehensive Pharmacogenomics Panel, which involves these 14 genes that we'll start with return for. Now, phase 2 on the data, these pipelines, there's 3 pipelines shown here. The top 2 are clinical. One is getting the result to patients. I'm showing a picture of PDF, but that's just the pretty visual. We're really focused on discrete results, so everything comes back by HL7 workflows and discrete results to the medical records at PIT and within UPMC, Epic, and Cerner. We also have analytic dashboards. This is awesome for trying to understand the associations with clinical value endpoints that UPMC consistently tracks. And the genomic data can be fed into there to see are patients that are carrying X variant or this metabolizer status, what are their care trajectories, how often do they come back to the emergency room, how often are they seeing their primary care docs, how often are they getting things filled in pharmacies. We have a unique, comprehensive look at the entire ecosystem, and we think there's value of understanding those care trajectories as data is returned to those patients. On the left-hand side is the research-based workflow. We are storing everything as discrete data as VCF, which allows us to use a number of tools, some of which you'll hear about shortly, I believe, to be able to do increased discovery and frequency analysis as we go out to more diverse cohorts. It also allows us to do some cohort discovery. So we're able to link investigators with cohorts that they think might be interested for further pharmacogenomic analyses. This is our engine for discovery I mentioned about sort of validating high-level targets. Those are the CPIC A's and B's on the left. And the ones that are lower, we believe we can spin in and move into the workflows as we demonstrate more value. For the EHR, this is what it looks like, and it's probably a little small for folks in the back, but the left-hand side I mentioned is all discrete results, so gene, diplotypes, and predicted phenotypes come from the lab. The lab's analysis stops at that. Our decision support picks up in the EHR after that. So we add on the recommendations, interruptive alerting within the electronic health records. And again, if you want to see the report, it's available, but we have open rates that are very low for that because folks are looking at the discrete results specifically. On the research side, this is a screenshot of our Cohort Explorer. We're able to look by ICD-10 and 9 and 10 codes, medication exposures, and it tracks some simple things like age of the population, what are the top 10, 20 medication exposures, and able to do analyses to try to link the two of them together. Just to close out here, looking at the percentages, of our early analysis is work by Dr. Kreiders, who are here in the front row. Looking at the analyses of exposures to folks where there were linkages between genotype and phenotype, as well as with opportunities for prescribing, and the rates are high, 38 to 50%, depending on what cohort we look at, allows us to see these data, and this is analogous, similar to what we're doing in seeing in other cohorts across the country. I think last slide here is on return of results. So we have a wonderful primary care precision medicine clinic where patients can come in and get direct pharmacogenomics care, as well as see the pharmacogenomics service on the inpatient side of things to support providers who need to make these recommendations. And then finally, we're starting to work with our portal and other solutions for getting results deliberately out to patients. And I think I actually have one more slide. The final plug I'll say, is that education is really important, and we're focusing on that here in Pittsburgh in scaling out training programs nationally to allow folks to learn how to do this, not only to enhance our own implementations, but also to understand how to best deploy in practices beyond from here. So with that, I'll stop and thank the large group. It's obviously not just me here in Pittsburgh. We have a very large team within the Institute, within CTSI, within pharmacy that helps us accomplish all this great work. So thanks.